So today we're going to be looking at the practical into diffusion. To do this, I have a small agar block, and inside this agar block is an indicator called phenolphthalein and an alkaline called sodium hydroxide. Phenolphthalein is pink when we have an alkaline environment. So we're going to look and see how does hydrochloric acid diffuse into this agar block. I can show you that very easily by adding the hydrochloric acid. So now we have hydrochloric acid diffusing through the agar. I'll bring it closer so you can see. Slowly you can see that the agar is turning colourless, and this is because the hydrochloric acid is diffusing into the agar block and it is neutralising the sodium hydroxide so that the pH changes, it rises up to 7 and the phenolphthalein indicator turns colourless. Now the question is, what about the rate of diffusion? How long does it take for this to happen? And what would happen if we had a different size agar block? So we're going to take a small agar block like this, and we're going to turn it into 3 different sizes. First of all, we're going to cut it in the middle so that we have a large piece of agar. Then we're going to cut the second piece of agar on the side, so we now have a medium piece of agar. And finally, again cutting in the middle, we have our third small piece of agar. The final piece of agar we do not need, so we are going to throw that away. What are we going to do with this investigation? Well, there's a number of different things that we can investigate, and on your worksheet we will be looking at the conclusions we can reach. Firstly, we know the dimensions of each agar block. They are 10 millimeters by 10 millimeters by 20 millimeters. The next one is half that size and half again, and you can look on your worksheet to see the dimensions. So we can see how does the volume affect the speed or the rate of diffusion. We could also consider how does the distance to the middle of the agar, how will that affect the rate of diffusion? And we could also think about the surface area compared to the volume. How will that affect the speed of diffusion? And we're going to do this very simply. I'm going to take each block in turn and put them into the test tubes that you can see here, the boiling tubes you can see here. Then I'm going to add an excess of hydrochloric acid, and I'm going to press start on the stopwatch. Your job is to watch for 3 stopwatches and watch for 3 agar cubes, and you have to write down on your worksheet on the OneNote the time when all the colour has finally gone. How long does it take for the entire pink colour to go and for the hydrochloric acid to diffuse through to the centre of the agar cube? So we're going to start our first one here now. So here you can clearly see the results of the experiment. We've got test tubes A, B, C, D with the size of the cubes that we had. The largest cube was A, the medium cube B, and the smallest cube was C. And there we can see the times that it took for each cube to go colourless. Cube A was our largest cube and it took 5 minutes 41 seconds to go colourless. In your table, in your worksheets as I'll give you later, it asks you to convert this into seconds, so that will be 641 seconds. Tube B took 340 minutes, which would be 220 seconds. And tube C, our smallest cube, took just 51 seconds. So what's our conclusions? What can we say? Well, clearly, the larger your cube, the bigger the volume, the more time it takes for diffusion to take place. It's a longer distance for the acid to reach the very centre of the agar cube and turn the alkali into neutral, so that the indicator changed from pink to colourless. Much longer distance. But there's other things going on here as well. Not only does a long distance slow down diffusion, but we also have to consider comparing the volume to the surface area. And in your worksheet they'll ask you to do a calculation where you compare the volume and the surface area to create something called the surface area to volume ratio. And when you do the maths, you'll find that much larger objects actually have a very small surface area compared to their very large volume. And this can affect the speed of diffusion. If you have a small surface area to volume ratio, diffusion will take a much longer time. Our smallest cube had the largest surface area to volume ratio, and so diffusion takes a much shorter time. And if you look in your body for organs like your lungs, like the placenta, the small intestine, they're adapted to have very fast diffusion, and one of their main adaptations is that they all have a large surface area compared to the volume, a large surface area to volume ratio. And that speeds up diffusion, it speeds up the movement of oxygen into your body or nutrients diffusing into your blood during digestion. So the last task you need to do now is to download the worksheet that goes with this practical, to complete and fill in the table using these results, follow the instructions and answer every single question. I hope you've enjoyed this experiment into diffusion.